Hello everyone, today we will talk about the gram negative and gram positive bacteria. So, gram positive and gram negative bacteria are classified on the basis of how they retain the gram stain. And this procedure of staining is called gram staining. This is also called as differential staining because in a population of bacteria, they can classify the bacteria into two different groups and we call them gram negative and gram positive. And this stain was named after the scientist who discovered it, and his name was Christian Gram, and he developed this in year 1884. He used crystal violet as primary stain, which is a basic and positively charged dye. Since it is positively charged, it's bind to negatively charged group present on cell wall of the bacteria. So this is the procedure, gram staining procedure. So we take the bacterial culture and first stain it with crystal violet, and then we add iodine, which is a mordant. Mordant means it helps to fix the crystal violet stain, and then we wash it with 95% ethanol. Ethanol act as decolorizer, and what it does, it actually, when bacteria is loosely bound with this crystal violet, ethanol can wash out all the stain. So after once it is after washing with 95% ethanol, we again stain it with safranin and another counter stain, because whatever bacteria who have already lost this stain, crystal violet stain, can now be colored with safranin. And when we observe under the microscope, we can see two different population of bacteria. One will be stained in purple color and another will be in red color. So bacteria which retain the crystal violet color are called gram positive bacteria, and who have lost this color and now they are stained with safranin and they appear red or pink in microscope, they are called gram negative bacteria. So why they are different? So answer is bacterial cell wall. So if you look at the, this is the structure of bacteria. So here this is cytoplasm, this is plasma membrane, and this is the outer layer, that is peptidoglycan layer. In gram positive bacteria, this peptidoglycan layer is very thick and it is about 20 to 80 nanometer of thickness. If you look here in case of gram negative bacteria, here it is cytoplasm, this is this white layer is periplasmic space. Periplasmic space is also present in gram positive bacteria but is not well defined as in case of gram negative bacteria. After periplasmic space, this is the peptidoglycan layer, which is very thin, which is 10 times less than the gram positive bacteria. It is only 2 to 8 nanometer. And this is outer layer. This is also the phospholipid bilayer present in gram negative bacteria, which is absent in gram positive bacteria. So what happens, since gram positive bacteria have multiple layer of this peptidoglycan cell wall, it strongly binds to crystal violet stain. But since in here in gram negative bacteria, this peptidoglycan layer is single layered, it loosely bound to a stain, and when we wash it with ethanol, it it loses all the stain. So that's why we see in gram positive we see the color of crystal violet, whereas in case of gram negative we see the color of safranin. Another difference between gram positive and gram negative is gram negative bacteria have two membranes. One is this cytoplasmic membrane, and here this is outer phospholipid bilayer present, and here they have lipopolysaccharide present which act as endotoxin. Why it is endotoxin? First, it is endo means it's integrated to the membrane. This is lipid and this is polysaccharide part attached to this lipid. And when this bacteria infect to host cell, and it is recognized when lipid is released, and it is recognized by, it binds to toll-like receptors of immune cells like macrophages, and then they activate the innate immune system. They release cytokines, they cause inflammation, fever, and they can also in severe cases can septic shock and also cause death. So this act as endotoxin. These are the examples of gram-positive and negative bacteria. So Staphylococcus aureus, Streptococcus pneumoniae are cocci, and Clostridium botulinum is bacilli. Similarly, gram-negative Escherichia coli is bacilli, Neisseria gonorrhoeae cocci, and Vibrio cholerae is commacept. Since in many textbook you find that they show the different shape of bacteria, like if it is gram positive they will show rod shape, then gram negative they will show the cocci shape. They are just representing this, but in actual scenario they can be of either shape. They can be cocci, they can be bacilli, they can be comma shaped. So this is the summary. First one, on the basis of a stain color, gram-positive bacteria retain the primary stain, that is crystal violet, so they look purple or violet. Gram-negative bacteria lose the crystal violet stain, so they capture the stain of safranin, so they look either pink or red. Gram-positive bacteria have very thick peptidoglycan layer, that is 20 to 80 nanometer, and that's why they hold the crystal violet stain very strongly, whereas gram-negative bacteria have very thin layer, only 2 to 8 nanometers, so they lose the stain. Outer membrane is absent in gram-positive bacteria and present in gram-negative bacteria. In outer membrane, lipopolysaccharide is present in case of gram-negative bacteria, and since outer membrane is not present in gram-positive bacteria, lipopolysaccharide is also absent. Teichoic acid is another difference which is present in gram-positive bacteria, but that is absent in gram-negative bacteria. Last one is endotoxin, which is absent in gram-positive bacteria because this lipopolysaccharide itself is acting as endotoxin. It is absent in gram-positive bacteria and present in gram-negative bacteria. So this was all about the gram staining, and basic differences between gram-negative and positive bacteria. Thanks for watching. See you in the next video.